We live in a market-dominated world. Markets do have a boundary of financial ethics. Yet the political and commercial factors are dominant factors in the modern world. At the end of 20th century, we realized that the financial advances are at the cost of loss of balance in nature. A threatening level of pollution and lost ecological harmony was a serious concern for modern world. Perhaps like sand we want to hold, yet slipping out from our hands. These advancements resulted into loss of balance of nature. Malaria has been a serious challenge for modern world, even today in 21st century. There were efforts to eradicate malaria by finding solutions like DDT and organochlorine, a chemical pesticide. With little success in handling the malaria issue, we eventually understood two things. One of them was the insects develop resistance against toxic chemical pesticides. Second was, toxicity of these chemicals accumulates into the food chain. The implausible solutions turned into a credulity Olympic, where the celebrated success of DDT resulted into a withdrawal. A pity withdrawal. The Silent Spring, written by Rachel Carson, was an eye-opener. The heavy cost of finding the industrial, science-based solutions resulted into many new problems. Yet today, malaria stands out to be a serious issue. Today in the modern world also, in the 21st century. Malaria is an infectious disease. It is caused due to infection of a protist, an eukaryotic microbe known as Plasmodium vivax, and there is one more species called Plasmodium falciparum. This belongs to kingdom Protista. Bacteria belong to kingdom Monera, while Plasmodium belongs to kingdom Protista. It has a well-defined nucleus and hence eukaryotic, but it is a microbe that is transmitted through insect vector and specifically mosquitoes. Mosquitoes are otherwise frugivorous, they feed on nectar. But during the breeding season, the female mosquitoes they become suddenly sanguivorous, means they feed on blood. They feed on blood of humans. So when they become sanguivorous, they insert the proboscis, the proboscis, into the skin of the host and reach the blood capillaries. They release an anticoagulant through that proboscis and there come out the plasmodial spores and those enter into human body. In circulation, they reach the liver. Liver has a dual supply of blood. One of them is the hepatic artery that carries blood to liver, and the second one is the hepatic portal vein that drains blood from stomach, spleen, and intestine, and that reaches back to liver. So it has a dual blood supply, profuse, rich blood supply. Inside the liver cells, this pathogen multiplies, and that results into necrosis of the cells there. Natural death of cells is called apoptosis, while death of cells due to some chemical factor, some infection, some detrimental kind of injury, that is known as necrosis. After necrosis of liver cells, the pathogen now circulates within the body and reaches the red blood cells. While invading the red blood cells, this plasmodium needs a cytoskeletal structure that helps to penetrate into the RBC. Plasmodia, they are classified into phylum Apicocomplexa. Apicocomplexa means those which have apical complex in a structure called apicoplast. This word apicoplast seems similar to the word chloroplast. The similarity is evident here, that is, just like chloroplast, apicoplasts also have two membranes. The first membrane that is found in apicoplasts, that is host-derived membrane during the process of endosymbiosis. Lynn Margulis, an American microbiologist, she gave this theory of endosymbiosis, means the organelles like chloroplasts or mitochondria, they are bacterial in the origin. They were bacteria which were engulfed by the larger cells, and hence during the process of that engulfing by larger cells millions of years ago, they were surrounded by the host cell membrane in which they entered, and that resulted into formation of a double membrane structure, outer host derived membrane and own native membrane, that is inner one. We will talk about this process in some more details in the next few parts, but right now uh, chloroplast and apicoplasts both are structures which are produced as a result of endosymbiosis. So this apicoplast, it has its own native DNA, just like chloroplast and mitochondria have, and it has its own protein synthesis machinery inside it, which is apart from the DNA present in the nucleus of this plasmodium. So there are three places where this microbe is likely to have DNA. One is its own nucleus, that is linear DNA, and other is its mitochondria and apicoplasts. Apical complex is a cytoskeletal structure. It is made up of some proteins called HSPs. These proteins belong to a special class of proteins known as molecular chaperones. Chaperones are essential in all cells because they shape the proteins. I usually compare teachers with chaperones, molecular chaperones. See, when there is protein synthesis, there are two subunits to the ribosome, a larger subunit and a smaller subunit. And this smaller subunit reads the code present on mRNA, which is made up of triplets, what we call as codons. The larger subunit that is above that, this larger subunit receives the transfer RNA, and this transfer RNA brings a specific amino acid at that location. Now, from ribosome, there emerges out a chain of amino acid residues which are linked by peptide bond. This chain enters the surrounding water. And you know, water is a polar molecule. Amino acids also have specific kind of polarity, few of them, and other constraints present in the cytoplasm, they help to fold this chain at a place. But this folding is not enough. Just like when we learn on our own, that learning is not very precise. But a teacher is the one who shapes these, who shapes our process of learning, who helps to make our learning more and more precise. Similarly, chaperones are structures which give proteins a perfect conformation. All biomolecules need to have a specific three-dimensional shape. This three-dimensional shape, or otherwise called conformation, this conformation needs to be extremely precise at nanoscale. It needs to have a very specific shape. For example, if you are holding a lock and if you try to put the key from other side, some other way, then it won't reach there. Okay, if you use some other key and that does not belong to the lock, and if you are trying to put it inside, that won't work. So there should be perfect complementarity between the lock and key so that that key can push the lever system inside the lock and it can open out that. Similarly, biomolecules needs to have a very complementary structure. May that be antigen and antibody, may that be a substrate and enzyme. There should be a complementarity between these shapes. And that precision in the shape of proteins is brought by these structures, what we call as molecular chaperones. If there is some misfolded protein, then inside the eukaryotic cell, there are small dustbins called proteasomes. That misfolded protein reaches that dustbin, molecular dustbin, and an enzyme protease that hydrolyzes the amino acids, and those are released back, recycled back into the cell. This is an interesting process. I usually compare teachers with these molecular chaperones. Just like a ribosome producing a chain, and in cytoplasm that polypeptide chain is getting folded itself, the process of learning is intrinsic. We learn every moment, but our learning becomes extremely precise when a teacher shapes it out. All right. So these in apical complex, apicocomplexa, this plasmodium, pathogen of malaria, there is a cytoskeletal structure called apical complex made up of some proteins which are called HSPs also, which surround the cytoskeletal structure there. And this malarial pathogen gets an ability to invade inside the red blood cells. So, this plasmodium has infected two hosts. Its life cycle continues through two hosts. First is mosquito and then the human host. So there are two hosts in the life cycle of plasmodium. But when in the body of a human host, this plasmodium harms it. It causes necrosis of cells in the liver. It causes destruction of RBCs as it multiplies into the RBCs. And this is also a kind of symbiosis. A symbiosis that is exactly opposite of mutualism, and this is known as parasitism. Plasmodium is a parasite of some cells in our body. It destroys, it harms those cells. In mutualism, host is benefited as well as the symbiont is benefited. In parasitism, the host is destroyed, it is at loss, and the parasite is at the benefit. So, we shall talk a few more details of malaria when we will start with the topic diseases. But right now, let us understand that there is one more type of symbiosis that is called parasitism. And now let us go to some botanical part. Ferns, pteridophytes. This is the second largest diverse class in the plants after angiosperms. In plant kingdom, the most diverse group is angiosperm and second largest diverse group is ferns. Ferns are of two types. The first type is megaphyllous ferns, means ferns having very large leaves, and ferns which have small microscopic leaves. They are called microphyllous fern. And here also we come to reference of the word mosquito anyhow. There is a type of fern called mosquito fern, or this is also called a floating fern known as Azolla microphylla, Azolla pinnata. There are various different species of this Azolla. In South Asia and Southeast Asia, in China and northwest of India, northeast of India as well, there has been a practice of using this floating fern in the rice fields during the cultivation. When rice grows, it requires large amount of nitrogen. The amount of nitrogen that needs to be fed to the crop of rice is almost 20% of its exact output. So it needs more nitrogen. Oryza sativa, rice. There are various different species and subspecies, some varieties. So this rice, in these rice fields, this fern that floats on the basal submerged part, and this is very rapidly growing fern. Almost within 2 days, the biomass of this fern that becomes 2.5 times or almost 3 times, it proliferically increases its biomass. And that is why its quantity increases when we are using it as a biofertilizer naturally. This Azolla, in one of its leaf lobes, which are in the form of a dorsal and ventral lobe, a pair, in one of these lobes there is a cyanobacterium called Anabaena azolae. This fixes atmospheric nitrogen and that serves as a biofertilizer, source of nitrogen, because rice needs a large input of nitrogen. So here is one more example of symbiosis. The symbiotic association of cyanobacterium called Anabina azole inside the leaf lobes of that floating fern, what we call as Azolla. And this is such a beneficial symbiotic combination, a mutualistic combination, that it results into such a excellent form of biofertilizer which increases the paddy yield by more than 50%, sometimes even exceeding 70%. And this has been a question in lot of MHCET exams in the past, and I suppose in NEET also. So Azolla is that biofertilizer. This biofertilizer is used specifically in the rice fields. So in today's lecture, we discussed the apicoplasts which are present in the malaria pathogen. Malaria pathogen itself as an example of symbiosis, but a parasitic kind of symbiosis. And the third example we discussed today is the Azolla cyanobacterium symbiotic association, Azolla anabina symbiotic association. Friends, thank you for listening me so kindly. This is Lakshmi Raman Oza, and you were watching SBTL webcasts.